The cell is the basic structural, functional, and biological unit of all organisms. It exists in different animals and plants to maintain their lives, especially in our human beings. However, around 100 million cells die every minute in different organs or tissues of human bodies throughout our lives. Fortunately, around 100 million cells divide to replace those dead cells in the same minute. There are three types of cell death, which are necrosis, autophagy, and apoptosis. In this video, I will just introduce cell apoptosis. I will describe morphological change of cells during apoptosis, mechanisms inducing apoptosis, and the implications of apoptosis in different biological processes. During apoptosis, normal cells undergo shrinkage and blubbing. Subsequently, the contents of cells collapse, and the cells degrade into fragments. At the same time, the apoptotic cells signal to the surrounding cells to engulf them. Finally, the fragments are internalized by the surrounding phagocytotic cells, such as macrophages. Apoptosis can be initiated through one of two pathways. They are intrinsic and extrinsic pathways. In the intrinsic pathways, the cell kills itself because it senses cell stress, while in the extrinsic pathway, the cell kills itself because of signals from other cells. In the intrinsic pathway, cytochrome C is an important factor inducing cell apoptosis. The cytochrome C is released from the mitochondria. It is regulated by different types of BCL2 family proteins. The BCL2 proteins can be activated by damage signals, such as death receptors, growth factors, phosphoinositide kinase, and p53. The first type of BCL2 family proteins is anti-apoptotic BCL2 proteins, such as MCL1. They act on the outer membrane of mitochondria to decrease permeability. Another type is pro-apoptotic BCL2 proteins, such as BIM. These make the holes in the outer membrane of mitochondria. The last type is BH3-only proteins, such as BID and BAD. These activate or inhibit the other two groups. Caspase-9 is a kind of initiator caspases, which induces intrinsic pathway of apoptosis. With the help of apaf1, which is an adapter protein, caspase-9 can be induced into dimer and cleave itself into active form. More specifically, the intrinsic pathway of apoptosis initiates when apaf1, an adapter protein, is induced to oligomerize into a heptamer by cytochrome C. The heptamerate complex is called apoptosan. The apoptosan also exposes its card domain, which binds the card domain of caspase-9. This results in recruitment of multiple molecules of caspase-9 to the apoptosan, inducing it to form active dimers. Activated caspase-9 then cleaves itself and is released to cleave and activate executioner caspases, which rapidly process the molecules that drive apoptosis. It's all over in a few minutes once the apoptosan has formed. The other pathway inducing cell apoptosis is extrinsic pathway. In this pathway, cytotoxic T cells come along and use their Fis ligand to interact with target apoptotic cells. Fis ligand uses death domain to bind the death domain of fat. After that, fat uses this effect domain to interact with the same domain of caspase-8. After recruited to the fat, caspase-8 undergo dimerization and become active. The activated caspase-8 cleave themselves and are released to activate executioner caspases. Actually, cell apoptosis is indispensable in embryonic development, such as digit formation. During early embryonic development, apoptosis occurs in the interdigital regions to make toes or fingers become apparent. Apoptosis plays a key role in selection of immune cells. Some immune cells recognize self-antigens during development. This will lead to autoimmunity. Therefore, these cells will undergo apoptosis to prevent from autoimmune disease.